infectious particle that requires a host to complete its life cycle. So a host like you or me. Well, HIV stands for Human Immunodeficiency Virus, and it is the virus responsible for AIDS. There are actually a lot of HIV drugs available now, but one of the problems with those drugs is that they are, first of all, very toxic, so they make people very sick when they take the drug. In addition to that, the virus is very, very tricky in that it can very quickly evolve resistance to the drugs, making the drugs no longer effective. And we both work on DNA and RNA aptamers. So what aptamers are very small DNA and RNA molecules that bind to a target protein. In our case, we work with the reverse transcriptase protein of HIV.
Basically, we block the enzyme from binding what it would normally bind in the cell by having it bind these RNAs instead. But in order to stop this enzyme from working, these aptamers need to bind to the enzyme, and they need to bind tightly. The idea being that, the better they bind to this enzyme, the better they're going to be able to inhibit it.
So my component of the research in our lab looks at these different aptamers and how they function to inhibit the virus. And I test each of the aptamers against the virus to see if we can stop the replication of the virus using the aptamer. Without a functional reverse transcriptase, HIV cannot complete its life cycle and is not infectious.
So my work has been focused on sort of understanding what structures these RNAs have and how this allows them to bind to this particular enzyme. We generate a pool of many RNA sequences. You can't, as a person, just look at those sequences and say anything meaningful, but we have software that can actually take these sequences and evaluate what structures are there. Some of the software we use basically can look at what these sequences have in common and infer from that what structures are present. We have this population of RNA molecules that bind to RT. But some of those RNA molecules can bind very tightly to RT, some bind kind of weakly. These aptamers need to bind to the enzyme, and they need to bind tightly. The idea being that, the better they bind to this enzyme, the better they're going to be able to inhibit it.
I think people tend to overemphasize HIV. So, people worry about getting HIV off of toilet seats and off of the gas pump, and it's really just more wimpy than people think that it is.